speaker is Alejandra Medina-Rivera. She's Associate Professor at the National Autonomous University of Mexico. During the last year, she has been focused on developing bioinformatics tools and strategies to study gene regulatory mechanisms. Today, she will talk about characterization, the effect of genetic variants within promoters with distal enhancer functions. I can't wait to learn more about this subject. So Alejandra, we are all ears.
Thank you very much for having me here. I am really excited to share my work in this platform. I will now share my screen. Hope that looks fine. So everyone's looking at the presentation and not the presenter's view. Am I right?
Yes, you're right.
Okay, thank you. Okay, so thank you for having me here. So I am at the National University of Mexico, as you already heard, but my campus is not in Mexico City, it's in Querétaro, which is a city in the middle of the country. And it's a recently formed research space. It's a really young institute. It has only 5 years. It was founded in 2015. And I like to show off that it's one of the few institutes that I know of where the majority of faculty are female. So I'm very proud to be part of this community, and I think it's a very research change community. So we really discuss a lot, and I really enjoy being part of it. So if you are interested on joining, please reach out to the different researchers. So today I'll talk to you about the rerulatory genome. And the rerulatory genome, as I see it in my research, is mostly composed by two proteins interacting with the DNA, the histones, that are the proteins where the DNA is wrapped around and that help to compress the genome and fit it into the nucleus, and transcription factors, which are proteins that bind specifically to certain places in the DNA that are marked by change of nucleotides. So that basically means words. So they look up for certain words in the genome and they bind to them. So thanks to these different regulatory interactions, our genome basically acts as a book of recipes, but you don't really need every recipe done at all times. So for example, you don't really need a turkey for Christmas this weekend, no? So you basically need just a normal barbecue. And that's how our genome works. And basically, we are able to have the same genome in all our cells, but our cells will make different things depending on where they are, if they are in the brain, if they are blood cell types, or they are in the kidneys, or they are bone. So that's super important because we have basically the same genome and we really make different things. And this is thanks to this regulation that tells us what genes are supposed to be on and which genes are supposed to be off, or which genes should be a little bit on or off, because there's a lot of modulation as well, because you do need some of the genes everywhere, but you don't need the same amount. So this is a brief schematic of how the regulatory genome is composed, and this is how what we have learned along many years of research. So we have learned that the histones, these protein balls where the DNA is wrapped that helps compressing into the nucleus, can have different post-transcriptional modification. And this modification, we have associated them to functions. So for example, if we see an acetylation and a methylation, we identify this region as an active enhancer, and enhancers are these long-range interacting genomes that basically depend on the DNA looping, and when the DNA loops, these enhancers that were far away now are really close to the promoter, and we have here the active promoter that also has in its histones specific modifications and that allow these regions to be open, because that is important. So if you have the DNA that is too compressed into the nucleosomes with the histone marks modifications, then it is too tight and the RNA polymerase cannot come in. But if you have it a bit loosened, then the RNA polymerase can come in and it can transcribe the genes. So actually, we also know there are certain modifications that have been related to the active gene region, so active transcription that is happening here. Or we also have some modification that have been related to latent enhancers, so some regions that are close, but that could be open pretty fast. And we also have some marks that have been related to repression. And one thing that I'm interested on as well is that there are these transcription factors that interact with these regulatory regions, either the enhancers or the promoters, and that help the RNA polymerase either are starting the transcription or repressing the transcription, because they can also have a repressive function. But as I told you, they like to bind to these specific motifs that I show here as a word motif. So that is a word in the DNA where this yellow transcription factor is bound. But you can also have mutations that will impede this binding to happen, because this word will no longer be recognized by the transcription factor. And this is important because now we have learned that we have a lot of variation in our genome that has been associated to either diseases or traits that are mostly in our known coding regions. So this is the 2012 schematics of the GWAS catalog variants. So those are our variants that have been associated or related to diseases or traits in humans. And as you can see in the different chromosomes, you see the different colors would mean that they are related to digestive systems or cardiovascular system or so on and so forth. And you can see that at the time, from the 70,000 reported variants, 93% of them were in non-coding sequence, which is usually related to these regulatory functions. And I just quickly show you the last figure from 2009, and you can see now that we have almost 160,000 variants that have been related to diseases or traits in humans, and 90% of them keep falling in these no-coding regions. So we really need to understand what these variants are doing in these enhancers and promoters. And this is very important also because I want to point out that we as biologists love categorizing things. So we really like to say this is an enhancer and this is a promoter, no? And this is a brief list of the first enhancers that were described in the literature. And as you can see, they come from different organisms, either various, human, mouse, even orchid. And you can see that these are very short, still short sequences that actually were not as far from the transcription start site. And that was one of the basic characteristics of enhancers, that they need to be long-range interacting regions that when the DNA loops, they touch the promoters, but actually the classic examples are really close to the transcription start site, so they're likely also promoters. So one of the things about categorization is that we have come up with a list of characteristics that should be present in promoters and enhancers. And for example, promoters should induce transcription of an heterologous reporter gene, while enhancers can have this long-range interaction and activate a promoter that is far away. We also have different properties in transcriptional initiation. For example, promoters usually only transcribe toward the gene that is downstream, while enhancers will have transcription that is divergent. So the RNA polymerase could sit there and transcribe to both sides indistinctly. We also have different ratios between these forward and reverse transcripts, and you can see that in enhancers it's equilibrated, while in promoters, well, the function is to transcribe their gene downstream, so you can see that there is a skewness in the ratio, and usually things go towards the gene. We also see more polyadenylated tails close to promoters, while we don't really see these in enhancers. Histone modifications are a bit different. Both have acetylation in the lysine 27 of the histone tree, but you can see that the methylation is different, and you see more trimethylation of lysine 4 in promoters, where you see monomethylation in enhancers. Moreover, we also see that in most cases, you see the RNA polymerase sitting there, which means that that is the reason we actually see all these divergent transcripts in enhancer, and well, promoters are there for the polymerase to sit and start transcription. We also see a difference in CPGI glands. While we see more in promoters, we see less in enhancers. So one of the questions is that, okay, so we actually see this list of classic enhancers that are too close to the transcription start site, that are likely being also promoters. So we wanted to know which of these promoters could actually have this enhancer function as well. And this is work done in collaboration with Salvatore Spicuglia at Aix-Marseille Université, at the TSEG, and it's also people from the University of Montpellier. And in my group, work has been mostly done by my student, Lucia Ramirez. And again, so we want to know whether a region, in this case promoter, is an enhancer or has properties to behave as an enhancer or not. So one of the developed high throughput techniques was StarSeq. This was published in 2015 by Alexander Stark's group. And basically what this assay does is that there is a vector that has a weak promoter, an open reading frame, and then you can clone the region that you want to question if it's an enhancer or not, and then a poly A site. So you clone this thing, and if it is an enhancer, it will help this weak promoter start transcription. So you will see RNA that is basically a self-transcription of this enhancer region over and over. So if it's an enhancer, you will see a lot of this RNA that self-transcribe. And one of the advantages of this technique is that you can clone many regions. So you can basically chop out your DNA and clone it into the same vector, and then transfect this library into a cell of interest, and then see which is the RNA that is being produced out of this, and align it to the genome. So you can identify which of these sequences self-transcribe and behave as an enhancer. And basically this is how it's look. So this is a library that was transfected into HeLa, and you can see here that there is a signal for enhancer in this region. And also one of the perks of this technique is that you can clone the exact same library into different cell types. So this is a figure from their paper for 2 different cell lines for Drosophila, and you can see that you detect different enhancers depending on the cell type. And that makes sense because you have different transcription factors, and transcription factors are key to activate these enhancer regions. So again, we wanted to know whether a region was an enhancer or not that could be activating promoters in long-range interactions. So in our case, the question is, do we have promoters that could also have these enhancers properties? So this was designed by Salvatore. So he designed this capture array for promoters annotated in RNA-seq, and he captured this genomic library with it. And these RNA-seq promoters were cloned into this assay, the StarSeq assay. So we were able to question whether this could self-transcribe, and that will answer our question of whether these promoters have enhancer properties or not. And this was transfected into HeLa and K562, and then RNA was sequenced. And through this assay, we were able to obtain certain promoters that show this enhancer property. And for example, in K562, we found 632. In HeLa, we found 493. And in total, we assayed 2,719 genes, promoters. And you can see that it's about 3% of the promoters in these 2 cell lines that show this promoter enhancer property. And we only have an overlap of 146, but this is actually something significant. So those could be a relevant promoters with this dual function. So one of the things we wanted to know, and this was already published, was whether the genes that are downstream, these promoters with enhancer potential, were actually transcribed or not. And we actually saw that they show a higher transcription than the general population of genes. So in red, I show you the genes that are downstream E promoter for K562 and HeLa. In brown, it's the ones that are common, these 146. And you can see in gray a sample of known E promoter associated genes. So these are genes that have normal promoters with no intention to also behave as enhancer. And you can see that we have higher expression of the genes that are downstream of these E promoters. So promoters that do have this enhancer potential. Well, the other question is that we wanted to know whether we also see a difference in the histone ratios, because as I told you before, histone marks have been clearly associated to this enhancer or promoter function. And one of the modifications that has been strongly associated to promoters is this 3-methylation of the lysine 4. So I wanted to know whether this acetylation versus methylation ratio was different in our E promoters, and we actually found out that yes. So I show you here the K562 signal of the ratio of the acetylation versus the 3-methylation. And these are the enhancers, the E promoters for only K562 and HeLa in blue and purple respectively. And you can see that in the K562 signal, we see a higher ratio of 27 acetylation over 3 methylation in the K562 specific E promoters, while this is reverse, sorry about that, in the HeLa specific E promoters, which means this is cell type specific. And the other thing is I wanted to know whether the genes that are associated to these E promoters had specific functions. So we did a gene enrichment ontology, and we saw that most of them are associated to immune response. So this is where the recent research of the lab starts. So we have these E promoters here shown in red, and we have these other promoters, which could be the ones that are the target of this enhancer function. And you can see that we propose that this is a way to quickly turn on many genes. So you have your enhancer, promoter enhancer that will turn on the gene downstream, but that will also interact with the other promoters that are nearby and turn them on. So this will make the reaction much more efficient, which makes sense because they seem to be associated to immune response, which is something that you want to do quickly. So here, for example, if you have a mutation in this E promoter, you would actually end up hitting also the other promoters, because now you no longer will be able to recruit quickly these transcription factors. Hence, you will probably lose the activation of other regulatory mechanisms. So we wanted to know which variants could be associated to these enhancers with promoter function in our HeLa and K562 sets. So for this, we look first for the transcription factors that are enriched in this region. So we wanted to know which are the regulatory mechanisms that are at work in these places. And then we wanted to know which are the genetic variants that we could find in these regions. And later on, we want to assess whether this variant could be affecting transcription factor binding. So first, which are the regulatory mechanisms affecting E promoters? And again, we wanted to look for transcription factor bindings and enrichment. In this case, you can see here HNF6, so this is the crystal where it binds, and this is the word where it likes to bind. So what we did is that we took the promoters with the enhancer function, and we scanned them using the motifs with from JASPAR 2020. So basically, we look for the number of sites we could see in our active E promoters versus a control set of promoters with no enhancer function. And we found that there were 12 transcription factors enriched in HeLa and 140 transcription factors enriched in K562. Out of these, 147 are unique transcription factors. And I show you here only the top 10 transcription factors enriched in the two cell types, because otherwise you couldn't see anything. And particularly, we saw that there is an enrichment in both cell types for CREB5, which is a transcription factor that has been related to inflammatory signaling. And we also see YY1 that recently has been associated to looping. So it's actually thought that it has a similar function as CTCF to help loop the DNA. But moreover, it seems to be more related to promoter enhancer interactions rather than just stats. That is what CTCF has been related on. So we found this to be very interesting because we do see that YY1, Y12 are enriched in both cell types. And we confirmed this also taking data from REMAP. REMAP is a database from ChIP-seq experiments. So this is no longer motifs. This is counting the number of sites discovered by ChIP-seq for the different transcription factors in HeLa and K562. And we're able to recover YY1 in both cell types and CBP again. So we found this to be very interesting, and this confirms our observation based on motif search. And now the next step is that we needed to know, so we now have the transcription factors, we have the motifs that could be of interest, and now we need to look for the variants that fall within these regions that could be disrupting transcription factor binding. So which are these genetic variants? So we look up in DBSNP, that is one of the major databases for variations. The GWAS catalog that, as I told you, has this annotation of variants that have been related to traits or diseases. We have ClinVar, that is another database where annotations are done based on the probable medical relevance of the variants. And we have GTEx, that is a database of EQTL, so variants that have been associated with gene expression. And here I show you, well, the number of variants that we were annotating in the whole genome. And we also took into account variants that are in linkage disequilibrium with any of these. And we found that there are 1,677 variants that fall within the promoters of HeLa, and 2,464 variants that fall within the promoters of K562. And out of them, more than 100 are annotated in GWAS, 4 GWAS traits in both cell types. And we have some that have been related to ClinVar and to GTX respectively. And so now we have the motif. So now we have the words that could be hit by these variants, and we have the variants that fall within these E promoters. And so we want to assess whether these variants could be disrupting the transcription factor binding site. And this is important because genetic variants in gene regulatory regions can cause many diseases because they disrupt regulation and hence will impede or enhance the transcription of certain genes that should or shouldn't be expressed. And this can also be informative of regulatory mechanisms that are relevant for certain diseases. So again, we want to know whether a variant could be affecting transcription factor binding and hence affecting gene expression. So we developed a tool that is called Variation Scan that is part of the regulatory sequences analysis tools. And this was work led by my bachelor student, Walter Santana, who's now also looking for PhD opportunities. So if you are interested. So this tool is able to take any motif. So here's one example. And it's a flexible tool because also it can take any variant set. Sorry?
5 minutes, please.
Okay, yeah. So, okay, so this, so you can see now that through this tool, you can assess the effect of these variants. And I'll go a bit faster. Sorry, we thought I had 20 minutes and 10 questions. So we have here 25 variants that are likely to affect binding of cystic transcription factor in HeLa. And we do see an enrichment of GTEx, so these QTLs, within this set. And we have 230 variants that are likely to affect 66 transcription factors. And we see an enrichment of GTEx QTLs and also of GWAS variants within the K562 set. And we also wanted to see whether we could see an enrichment of certain diseases from taking the GWAS catalog annotation of experimental factor ontology. So we took the variants from HeLa and K562 that were in the GWAS catalog, and we found 37 diseases and 202 diseases. And we could see an enrichment of diseases related to immune functions, so like lymphomas and leukemia, and also other cell response as bone marrow neoplasm. And this is one example. So this variant is within the ANI promoter in K562 upstream region that is related to this tumor necrosis factor receptor, and is annotated as being benign in ClinVar, and it has been associated to the expression of a gene that is close by, LTBR, in whole blood from GTEx, and it's in LD with a variant that has been associated to changes in eosinophil count, so again immune disease, and it's predicted to affect the binding of IRF1. And you can see here, there is the variant here in the locus for this tumor necrosis factor receptor 1A. And here you can see the variant that is affecting this change. So we actually pass from a site that is considered to be of high thermodynamic stability to one that is low. So that is the effect of this IRF1 binding site. And you can see that we have the prediction of looping to this LTBR gene, that is the gene that has been associated to be changing expression based on this EQTL in GTEx. So in summary, high throughput techniques can be used to identify long-range regulatory sequences. We have several functions in these sequences. We can have promoter with enhancer function. These variants that are within E promoters can be related to human diseases, and variants within E promoters can potentially affect regulatory interactions. And further on, we want to try to relate diseases to these E promoters, and we are now going through experimental validation of some of the variants we found. So I want to acknowledge my lab, particularly Lucia Ramirez, that did most of the work that I show today, and the funding agencies that allow my lab to go on. And since you're already here, I want to invite you to also join ICB Latin America Soy Bio BioNet Mex Symposium 2020, And well, it's going to be virtual at the end of October. So if you're interested, I hope that I'll see you there. Thank you for your attention.
Thank you, Alejandra. We still have some minutes for questions. I will go with the first one. Zulia Fernandina Nieves Lopez asks, have you found a functional enhancer near or in region corresponding to transposable elements? If it's yes, do you think could have some biological advantage?
Well, in this work, we only focus on promoters, which are not particularly close to transposable elements. And we haven't really looked profoundly into the ones that could be, if they have anything particular. In general, transposable elements have been related to binding of zinc fingers. So I do think that there is an interesting question there, but we didn't look into that.
Okay, the last question. Does each promoter share sequence features with enhancers? In particular, are there shared motifs between e-promoters and enhancers?
That is a very good question. We haven't really looked into that either because it's really hard to establish a set of positive enhancers in the particular cell types. Now, with the new ENCODE, actually, there is a new set of golden standard enhancers in these two cell types that we like to check on as well and compare them.
Okay, thank you again.